Hello everyone and welcome to the new session of the lecture series. And today we'll be revising the topic photosynthesis in higher plants. Alright, so we have already discussed some of the topics in the previous lecture. Okay, so we were discussing about the earlier experiments and then we discussed about the mechanism of photosynthesis, photophosphorylation, what are the types of photophosphorylation, and then we also discussed about the photosynthetic pigments and all. Okay, and the structure of chlorophyll. And now today in this lecture, we'll be discussing about all the cycles, okay, all the pathways today, okay. And then we'll be discussing about the internal and external factors that affect the rate of photosynthesis. So let's start with the dark phase, okay, also known as the dark reaction, all right, also known as the biosynthetic phase, all right. So dark reaction is also known as the biosynthetic phase, light independent reactions, Kelvin cycle or C3 cycle. So it is the most common pathway for carbon dioxide fixation in plants. Alright, and what is C3 cycle? It is the biochemical reaction that takes place in the stroma of the chloroplast. Now what happens during the C3 cycle? Atmospheric carbon dioxide is converted into organic compounds, alright, and mostly or primarily glucose is formed. Okay. Other carbohydrates are also formed, such as cellulose is also formed, okay, and starch is also formed, but majorly glucose is formed. Now what happens here? The products of the light reaction, which are ATP and NADPH, are used for the synthesis of carbohydrate during the C3 or the Kelvin cycle. Okay. Now the enzyme involved here is rubisco. Okay. What is the full form of rubisco? Ribulose 1,5-bisphosphate carboxylase oxygenase. Okay. It has both carboxylase and oxygenase activity. Alright. And what does this enzyme catalyze? Rubisco. So rubisco catalyzes the reaction in the C3 cycle. Okay. If we are talking about the C3 cycle, the rubisco catalyzes the reaction between carbon dioxide and a 5-carbon compound known as RUBP, ribulose 1,5-bisphosphate. Okay, now let's talk about the Kelvin cycle in detail. Okay, so Kelvin cycle is divided into 3 distinct phases, or it occurs in 3 phases. First is the carboxylation phase, in which the carbon dioxide is fixed from the inorganic form to organic form. All right. And then second is the reduction phase, also known as the glycolytic reversal, means it is the reverse of the glycolysis pathway seen in respiration. Okay. Third is the regeneration phase, where what happens? There is regeneration of ribulose 1,5-bisphosphate takes place, okay, which ensures the continuation of the cycle. Alright, so some of the examples where C3 cycle takes place, okay, so these are the plants: rice, wheat, oats and rye. In these plants, C3 cycle occurs. Okay, now let's talk about the first phase known as the carboxylation phase. So what happens here? The carbon dioxide molecules are added to a 5-carbon compound RUBP, okay, to generate an unstable 6-carbon compound. Alright, so initially an unstable 6-carbon compound is formed. So this you have to remember. And this unstable 6-carbon compound dissociate into, okay, 2 molecules of 3-phosphoglyceric acid. Okay, 3-PGA. So this is the reaction here. Okay, RUBP combines with carbon dioxide in the presence of enzyme rubisco, give rise to 3-phosphoglyceric acid. Okay, now why I have written 6 here? 6 molecules of RUBP combined with 6 molecules of carbon dioxide. Why? Because what happens in C3 cycle? There is a formation of glucose molecule. And what is glucose? C6H12O6. Okay, means 6 carbon are involved here. Okay, glucose contains 6 carbons. So 6 carbon dioxide has to be fixed for the formation of 1 molecule of glucose. Okay, that's why we are writing 6 molecules here. So if we are using 6 molecules, of carbon dioxide, 6 molecules of RUBP will also be required. And what will happen? Then 12 molecules of 3-phosphoglyceric acid will be produced, because 3-phosphoglyceric acid is a 3-carbon compound. Okay. Here RUBP is a 5-carbon compound and carbon dioxide contains 1 carbon. Alright. So here 12 molecules will be formed. Now next is the reduction phase, also known as the reverse of glycolysis. So here what happens? There is a conversion of the 3-phosphoglyceric acid into 1,3-bisphosphoglyceric acid. Okay. So what happens again? Now 12 molecules of 3-PGA were synthesized in the carboxylation phase. Now what happens? 12 molecules of 3-PGA utilize 12 molecules of ATP to form 12 molecules of 1,3-bisphosphoglyceric acid. Okay. So now phosphorylation is taking place here using ATP and 12 ADP will be produced. Okay. Now another reaction takes place in the reduction phase. So what happens here? Now 12 molecules of 1,3-bisphosphoglyceric acid is formed. Okay. This utilizes 12 NADPH2. Okay. And what is formed? 12 molecules of 3-phosphoglyceraldehyde. Okay. That's why it is known as the reduction phase. Here what is happening? The acidic group or the acid group is converted into aldehyde. Alright. So this happens during the reduction phase, and what is utilized? ATP is utilized and NADPH is also utilized. Okay. Now finally 12 molecules of 3-phosphoglyceraldehyde are produced. Out of these 12 molecules, only 2 molecules of 3-phosphoglyceraldehyde are used for the formation of glucose. Okay, now you can see glucose is synthesized. Okay, that's why it is known as the reverse of glycolysis. Okay, in glycolysis, glucose is broken down, whereas there is synthesis of glucose here. So it is an anabolic process. It is an anabolic process, means complex compounds are formed from the simple compounds. Now, next is the regeneration phase. Okay, third phase. Here what happens? Now, 2 molecules of phosphoglyceraldehyde were used for the formation of glucose. Now, remaining 10 molecules are recycled to regenerate 6 molecules of RUBP. So, okay, and what happens? 6 ATP are used here. Alright, so now you can see in total 18 ATP are used. Alright, 12 in the reduction phase and 6 in the regeneration phase, and 12 NADPH2 are also utilized. Okay, for fixing how many? For fixing 6 carbon dioxide, or you can say for the formation of 1 molecule of glucose. 1 glucose molecule requires 18 ATP and 12 NADPH. Okay, so this is the input and output of the chemical cycle. Input was 6 carbon dioxide, 18 ATP and 12 NADPH. Output is 1 glucose molecule, 18 ATP and 12 NADPH. Now these are the steps involved in the Kelvin cycle. Okay, first is the carboxylation phase. Here what happens? Carbon dioxide will combine with RUBP. Okay, so carbon dioxide first combines with or mixes with water. Okay, and form carbonic acid, HCO3. And this HCO3 can then combine with RUBP. Okay, this is known as the carboxylation phase. And what will happen? 3-phosphoglyceric acid is produced. Okay, 3-phosphoglyceric acid and 3-phosphoglycerate is same. Okay, and then we have the reduction phase. Okay, reduction phase, what happens? Ultimately, 3-phosphoglyceraldehyde is produced. Okay, triose phosphate they are writing. Triose phosphate means a organic compound which contains 3 carbon. Okay, triose means 3 carbon. So after the reduction phase, we know what is produced. 3-phosphoglyceraldehyde was produced at the end of the reduction phase. Okay. Then what happens? In the regeneration phase, RUBP is regenerated. Okay, RUBP is regenerated, and this ensures the continuation of the cycle of the C3 cycle. Here again, coming back to the reduction phase, ATP and NADPH were used. Okay, and ADP and NADP plus are reduced. Okay, so this was the steps in the Kelvin cycle in short. Okay. Now, what is the significance of the C3 cycle? It assists in the plant growth. Okay, the glucose and the other carbohydrates produced are used for various metabolic processes such as the cell growth, cell repair and reproduction. Okay, and second significance is it supports life on Earth. Okay, how? By providing energy in the form of organic matter. Okay, that sustain all the heterotrophic organism. Heterotroph, which feeds on what? Producers. Okay, so plants are the producers and heterotrophs feed on these producers, these photosynthetic organisms, plants. Okay, and plants produce organic matter which are eaten up by the heterotrophic organisms such as deer or cow. Okay, now this was the end of the C3 cycle. Now let's start with the C4 cycle, which is also known as the Hatch and Slack pathway. So what happens? The plants that are adapted to or that live in or that reside in dry tropical regions, they show this C4 cycle. Okay, so C4 cycle is also an alternative photosynthetic pathway. Alright, seen in the plants adapted to dry tropical regions. So we can also say that C4 pathway is an adaptation that allows these plants to overcome the inefficiencies of the Kelvin cycle under high temperature and intense light conditions. Okay. Now what happens in C4 cycle? Carbon dioxide fixation takes place twice. It occurs 2 times. Okay, one in mesophyll cell, one in bundle sheath cells. We'll talk about this, okay, in detail. And then what we have, C4 cycle. What are the examples of plant in which C4 cycle is seen? It is usually seen in the members of the monocots. Okay, grass family. Monocots are usually, they are also commonly called as the grass family, or the Poaceae is also known as the grass family. Okay, and they also seen, C4 cycle is also seen in the members of Cyperaceae, for example, sugarcane, maize, sorghum, bajra and panicum. Okay, now C4 cycle also occurs in dicots, and the examples of Euphorbia, Amaranthus, Chenopodium, Boravia and Atriplex rosea. Okay, now if we talk about the C4 plant, they show a unique leaf anatomy or specialized leaf anatomy known as Cran's anatomy. Okay, so here is a diagram showing the Cran's anatomy. So what happens here? 2 types of photosynthetic cells are present here. Okay, there are 2 types of photosynthetic cell. One is mesophyll cell, another is bundle sheath cells. Okay, so what happens in Cran's anatomy? The mesophyll cells and the bundle sheath cells are arranged in concentric manner around the vascular bundles. Okay, so this is the vascular bundle here, the pink region. These are the mesophyll cells. Okay, the outer ones are the mesophyll cells. Then inner 2 mesophyll cells are the bundle sheath cells. Okay, bundle sheath cells, and they form a, okay, you can see a ring. They form a ring. These cells forms a ring, or you can say they form a wreath. Okay, crans. What is the meaning of crans? It means a ring or a wreath. Okay, cells are arranged in concentric concentric manner around the vascular bundles. Okay, now these vascular bundles are impervious to gaseous exchange. Okay, they are, you have to remember this, they are impervious to gaseous exchange. Okay, and what else is present in crayon's anatomy? Dimorphic chloroplast is present, another unique feature. So the bundle sheath cells contain agranule and large chloroplast, whereas mesophyll cells contain granule and small chloroplast. Okay, again one thing you have to remember, that the bundle sheath cells, they contain the enzyme Rubisco, which is involved in the C3 cycle. Okay, and the mesophyll cells, they contain another enzyme known as PEP carboxylase. Okay, so this enzyme helps in the primary carbon dioxide fixation. Alright, now let's study about the steps in the C4 cycle. So this is the first step, or the initial carbon. I've told you that carbon dioxide fixation or carbon fixation takes place twice. So this is the first carbon dioxide fixation, during which what happens? Okay, the carbon dioxide which is dissolved in water, okay, it combines with the phosphoenol pyruvate. So instead of RUBP, we have a 3-carbon compound here known as phosphoenol pyruvate. Alright, so this is the initial carbon dioxide fixation. You can just write carbon dioxide combines with phosphoenol pyruvate. Okay, so this has 1 carbon, phosphoenol pyruvate has 3 carbon, and what is formed? A 4-carbon compound known as oxaloacetate is formed. Okay, here you can see first carbon dioxide fixation. Okay, here you can primary carbon dioxide fixation here leads to the formation of oxaloacetate. Now this oxaloacetic acetate is further converted into what? Malate here. It is converted into malate in the mesophyll cells. Okay, so these are mesophyll cells. Okay, and then these are the bundle sheath cells. So oxaloacetic acid cannot directly enter the bundle sheath cells. It cannot enter. First it is converted into malate, and then malate is transported into the bundle sheath cells. Okay, so this is the second step. Okay, transport of a 4-carbon compound. Okay, here I have written oxaloacetate is further converted into malate and then transported to bundle sheath cells. The next step is the decarboxylation step. Okay, during decarboxylation, what happens? So we have malate in the bundle sheath cells now. So this is malate. It undergo decarboxylation. Okay, decarboxylation means release of carbon dioxide. Okay, or loss of a carbon. Okay, this results in the release of carbon dioxide, and a 3-carbon compound is formed now known as pyruvate. Okay, malate was a 4-carbon compound. After decarboxylation, a 3-carbon compound, pyruvate, is formed and carbon dioxide is released. And this carbon dioxide is utilized in the C3 cycle or the Kelvin cycle for the production of carbohydrates. Okay, here what will be produced after C3 cycle? Carbohydrates. Alright, now this pyruvate which is formed, okay, this pyruvate, now it transported back to the mesophyll cells. Okay, here you can see the pyruvate. This pyruvate is transported into the mesophyll cells again, and then this pyruvate is used for the regeneration of phosphoenol pyruvate, thus ensuring the continuation of the cycle of the C4 cycle. Okay, so here I have written in bundle sheath cells, decarboxylation takes place where? If someone asks you, in bundle sheath cells. In bundle sheath cells, malate is decarboxylated to and forms pyruvate, and carbon dioxide is released. Alright, Then what happens? The released carbon dioxide enters the Kelvin cycle and leads to the formation of carbohydrates. Now, what is the significance of C4 cycle? It helps in the reduction of the photorespiration. We will study about this process, photorespiration. So it is a wasteful process usually, which leads to the reduction in the photosynthetic rate. Okay. Photorespiration leads to reduction of the photosynthetic yield also, you can say. Photosynthetic yield. Okay. So C4 plants are adapted to higher temperature and all. Okay. Because of this adaptation and because of the presence of Kranz and ATME, there is reduction of photorespiration. So in Kranz and ATME, you have seen there was in C4 cycle, you have seen that there was a spatial separation of what? There is a spatial separation of carbon fixation. Okay. Spatial separation. Okay. One was taking place in the mesophyll cells and another carbon dioxide fixation was taking place in the bundle sheath cells. So this is known as the spatial separation of carbon fixation. Alright. Now another significance of C4 cycle is it increases the photosynthetic yield of the C4 plant. Okay. How it increases the photosynthetic yield? By reducing the respiratory pathway. Okay. And third is the significance is enhances the ability of C4 plant to thrive in high temperature and intense light conditions. Okay. So what happens? There is a prevention of water loss also in C4 plants. Okay. And there is an increase in photosynthetic yield also. Now let's talk about the carboxylase oxygenase activity of RUBP. So what happens here? We have already know that the Rubisco shows two kinds of activity. First is the carboxylase, second is oxygenase. Okay, carboxylase means it can bind to carbon dioxide. Oxygenase means the active site of the Rubisco can bind to the oxygen molecule. Okay, now let's talk about this. Relative concentration of oxygen and carbon dioxide determines which of the two will bind to the enzyme Rubisco. Now, another fact is that Rubisco has a much greater affinity for carbon dioxide when carbon dioxide and oxygen is nearly equal. Okay. Now, let's study about the conditions when oxygenase activity is favored. So, oxygenase activity is favored at high oxygen and low carbon dioxide concentration. Alright. Or low carbon dioxide to oxygen ratio. Okay. Then oxygenase activity is favored at higher temperature and very high light intensity. Okay. So if these three conditions are present, then oxygenase activity will be favored, means Rubisco will bind to the oxygen. Okay. Now this is the figure showing you the conditions required for the Rubisco for photorespiration and the Kelvin cycle. So at high temperature, low carbon dioxide to oxygen ratio and high light intensity, photorespiration is favored. Whereas in cooler temperature, high carbon dioxide to oxygen ratio and low light intensity, Kelvin cycle is favored. Now let's talk about the photorespiratory pathway, also known as the C2 cycle or the glycolate pathway. Alright, so here you can see three organelles are present: chloroplast, mitochondria and peroxisomes. Okay, so in C3 plants, some oxygen bind to Rubisco, thus decreasing the photosynthetic efficiency. Okay, and photorespiration, another fact is that involves the coordination between three organelles: chloroplast, mitochondria and peroxisome. So here you can see in this diagram, Rubisco shows oxygenase activity means now RUBP will bind to oxygen. Okay, instead of carbon dioxide And what will happen? 2 Pg is produced, means 2 phosphoglycolate is produced, which is converted into glycolate. Now, this glycolate which is produced enters the peroxisomes. Okay, then glycolate is converted into glyoxylate. Glyoxylate is converted into glycine. Now, this glycine enters mitochondria. Okay, now glycine is then converted into serine within the mitochondria. Okay, here 2 molecules of glycine are used. Okay, and then it is converted into serine, which is a 3-carbon compound. Okay, glycine is a 2-carbon compound. Alright, and there is a release of carbon dioxide here. Okay, so initially the carbon dioxide which was fixed, okay, in the form of phosphoglycolate is released here. Okay, that's why it is a wasteful process. There is a loss of photosynthetic yield. Okay, so here the carbon dioxide was fixed, but here there is decarboxylation and release of carbon dioxide. Alright, then this serine enters again peroxisomes. Then this serine is converted into glycerate. Now glycerate enters the chloroplast. Okay, and then glycerate is further converted into, or this glycerate then enters the C3 cycle. Alright, so this was about the photorespiration in brief. You can see there are 3 organelles are involved. Okay, and the initial product is a 2-carbon compound. So this glycolate, okay, phosphoglycolate, this is a 2-carbon compound. Okay, another thing I would like to mention here that Rubisco RUBP is a 5-carbon compound. Okay, so initially 3-phosphoglyceric acid is produced, which is a 3-carbon compound, and along with this a 2-carbon compound phosphoglycolate is also produced. Okay, phosphoglycolate, okay, which is a 2-carbon compound is also produced. Okay, then phosphoglycolate is converted into glycolate, and the rest you can see in this diagram. Now, what are the disadvantages of photorespiration? First is photorespiration decrease the photosynthetic efficiency. Okay, now there is neither synthesis of sugars nor of ATP and NADPH. So it is a wasteful process. Third is results in the release of carbon dioxide with the utilization of ATP. Okay, ATP is not produced, instead ATP is utilized. Alright, now let's talk about the CAM pathway. So CAM pathway is an adaptation to the arid climate. Okay, means dry climate. Examples are some succulents and some epiphytic plants show the CAM pathway. So here you can see the temporal separation of the carbon dioxide fixation, means carbon dioxide fixation is separated by time. Okay, so there is a temporal separation of the biochemical reactions that help in reducing the water loss through transpiration. So now crystallization acid metabolism takes place, okay, in 2 phases. First is the night phase. Okay, during night what happens? The stomata open and allow the gaseous exchange. Carbon dioxide fixation takes place at night. Okay, and carbon dioxide is stored as organic acid, malic acid. Okay, which acid? Malic acid, which is a 4-carbon compound in the vacuoles. Okay, again, so what happens here? Carbon dioxide combines with phosphoenol pyruvate to form oxaloacetic acid. This oxaloacetic acid is converted into malic acid. Okay, and this malic acid is stored in the vacuoles in the night time. Okay, enzyme involved is PEP carboxylase. Now what happens during the daytime? During the daytime, stomata remain closed. Okay, and the malic acid comes out from the vacuoles and is decarboxylated to form pyruvate, and carbon dioxide is released as a byproduct. Here what happens? Now this carbon dioxide which is released enters the C3 cycle, and the enzyme involved we know is the Rubisco enzyme, okay, which is involved in the C3 cycle. This is the diagram showing the CAM pathway. Here are some of the plants showing the CAM pathway. Here you can see carbon dioxide is present here. Okay, this carbon, okay, it starts from here. Carbon dioxide combines with the carbon dioxide. All right, and oxaloacetic acid is produced. Which is then converted into malate, which is stored in the vacuoles. And during the daytime, there is decarboxylation, release of carbon dioxide, and this carbon dioxide enters the CAM pathway. Now let's talk about the factors affecting the photosynthesis. Some are the, okay, so there are internal factors and external factors which affect the rate of photosynthesis. So let's talk about the internal factor first, also known as the plant factors. So these are number, anatomy, age, and orientation of the leaves. This affect the rate of photosynthesis. And then we have internal carbon dioxide concentration also affect the rate of photosynthesis, and the amount of chlorophyll also affect the rate of photosynthesis. Okay, more the chlorophyll, more will be the photosynthesis. Internal carbon dioxide, more will be the internal carbon dioxide concentration. Rubisco can easily bind to the carbon dioxide and follow the C3 cycle, all right, instead of the photorespiratory pathway. And the number, anatomy, age, as well as the orientation of the leaves also determine the rate of the photosynthesis. All right, we have already discussed about these factors in the previous lecture in detail. Okay, now let's talk about the external factors. So first is light. So if we talk about light, there are three parameters. Okay, first is the light intensity, light quality. Third is the duration of the light. Okay, and light is usually a limiting factor. Why? Because light saturation occurs at 10% of the full sunlight. Okay, now let's talk about the light intensity. So at lower light intensity, a linear relationship exists between the incident light and the carbon dioxide fixation rate. Whereas at higher light intensity, reduction in photosynthesis takes place. Why? Because there is a breakdown of the chlorophyll molecule. Or you can also say there is photooxidation of the chlorophyll molecule. All right. Okay, so this is the graph showing the effect of light intensity on the rate of photosynthesis. Here you can see the curve here. Here we have the light intensity, and this is the rate of photosynthesis here. Okay. Now you can see rate of photosynthesis initially increases as the light intensity increases, and then it becomes stabilized. Okay, at higher light intensity. Okay. So what happens at higher light intensity? There is photooxidation or breakdown of chlorophyll. That's why later on, what will happen? The photosynthetic rate will decrease. Okay. Now next is light quality. So if we talk about light quality, we talk about different kinds of light. Okay, light we know can be divided. Okay, if divided into the components, we have red light, blue light. Okay, we have the VIBGYOR. Okay, so red light and blue light, it results in the highest photosynthetic rate, whereas blue and the red light promotes the stomatal opening. Okay, we know chlorophyll absorbs maximum in red light and blue light. That's why highest photosynthetic rate. All right. Now let's talk about the green light. Green light induces the stomatal closure. Okay. If stomata is closed, there will be no gaseous exchange. So it will affect the rate of photosynthesis. Now again, blue light enhances chloroplast development. Okay, if the chloroplasts are developed, it will enhance the rate of photosynthesis. And another fact is that the combination of red, blue and green light can lead to the increased leaf area expansion. Okay, if the leaf area is increased, then what will happen? It will absorb more sunlight, and this will lead to the increase in the rate of photosynthesis. Now let's talk about the duration of the light. Now longer photoperiods provide more opportunities for light absorption and energy capture. Okay, if the plant is exposed to longer day length or longer photoperiod, then this will lead to more energy capture, and this will lead to the increase in the rate of photosynthesis. Okay, and we know that if you talk about photosynthesis takes place in two phases. First is light dependent, another is dark phase. Okay, so most of the reaction in the light dependent phase of photosynthesis require continuous exposure to light. Now let's talk about the concentration of carbon dioxide. Increase in carbon dioxide concentration up to 0.05% can cause an increase in the carbon dioxide fixation, but beyond this level it can be damaging. Okay, and this is a fact here. The C4 plant shows saturation at about 360 microliter per liter of carbon dioxide, whereas C3 plant shows saturation at levels that are beyond 450 microliter per liter of carbon dioxide. Okay, so this shows that C4 plants are more efficient, okay, than the C3 plants. They are more efficient. Okay, they have the adaptation known as the Cran's anatomy, okay, which helps in the reduction of photorespiration. Now let's again talk about the concentration of carbon dioxide. At lower light intensity, what happens? Both C3 and C4 plants, they do not respond to higher concentration of carbon dioxide. Okay, but at high light intensity, both C3 and C4 plants show increase in the rate of photosynthesis. Alright, so a graph looks like this. Okay, this one is straight. Okay, so this is at high light intensity. This is at low light intensity. Here we have the carbon dioxide concentration. Here we have the rate of photosynthesis. Okay, so here you can see at higher light intensity, the rate of photosynthesis increases, where at low light intensity, the rate of photosynthesis is a bit lower than, okay, both in C3 and C4 plants. Now let's talk about the temperature. Both light and dark reactions affected by temperature. The dark reaction involve a lot of enzymatic reaction and is temperature controlled. Okay. Now what happens? You know, the temperature affects the activity of enzymes. Okay, and enzymes, they work at optimum temperature. Okay, so what happens initially when we increase the temperature, then there is a lot of collision between the molecules. Then this leads to the increase in the rate of photosynthesis. Alright, and later if we exceed the temperature from the optimum, then what happens? Then it can denature the enzyme. At higher temperature, what happens? The enzymes involved in photosynthesis, they denature. Okay, so what will happen? This will lead to the reduction in the rate of photosynthesis. Alright, there will be reduction in the rate of photosynthesis and maximum. Okay, if we talk about the optimum temperature, at optimum temperature, what happens? Maximum rate of photosynthesis. Okay, there is a maximum rate of photosynthesis. Now, again, plants living in the tropical habitats have higher temperature optimum than the plants adapted to the temperate climates. Okay. Now, one more fact is that the C4 plants show enhanced photosynthesis at higher temperature, while C3 plants have a much lower temperature optimum. Now, next external factor is water deficit, okay, or water stress. So, water stress can cause the stomatal closure, thereby reducing the carbon dioxide availability. Moreover, the surface area and the metabolic activity of the leaf is reduced as the leaf wilt. Okay, all these factors contribute to the decrease in the photosynthetic rate. Now let's talk about the other factor, which is the soil. Okay, so water also helps to maintain the temperature required for the photosynthesis, and it also helps in the uptake of minerals and ions from the soil. Okay, so these minerals and ions are required for the process of photosynthesis. Now let's talk about the law of limiting factor. Okay, it is also known as the Blackman law. So it states that if the chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimum value. Okay, or you can also say that the rate of the process is usually determined by the one or by the only one major factor that is present at the suboptimal level, or you can say with the shorter supply. Okay, so this is known as the law of limiting factor. Okay, and what are the factors like carbon dioxide, okay, water, temperature, all the external factors. Okay, so the rate of process is determined by the factor which is present in the shortest supply, simply you can say. Now these are some of the references that I have used, NCERT book, and some of the papers and books and the books that I have used. Okay, you can refer to them. These are some sites from which images has been taken. So this was the end of this lecture. Thank you very much for listening.